Hey guys, how you all doing? I hope you all are doing good. So in the last lecture we studied about what is microscopes and the terms related to it. In this lecture we're going to talk about bright field microscopy, but before that we need to understand that is a type of light microscope and a compound microscope. What is a light microscope and a compound microscope? Simple. Microscope that uses light is called light microscope. So bright field microscope is a type of light microscope and the microscope that uses two lenses. Now in the parts section of bright field microscope, we're going to talk about the parts of microscope. So there will be two lenses, objective lens and one will be ocular lens. Okay. So that microscope uses two lenses. So such kind of a microscope that uses two lenses are called compound microscope. So bright field microscope is a light microscope because it uses light, and a compound microscope because it has two lenses to form an image. Okay. Simple. Other examples of light microscope will be dark field, phase contrast, fluorescence, all we're gonna study in different lectures. Right here, a compound microscope that uses objective as well as ocular lenses, the repetition of the same thing that I said. Okay, now the parts of the microscope. There are eight basic parts for bright field microscope or any light microscope. Those are light source. First is light source. Maybe it could be a bulb or it could be a sunlight that provides light to the microscope. The other one is diaphragm, which is helps in controlling the amount of light. It might be possible that the bulb has great intensity and we do not need that much of intensity. So we can control the amount of light, or maybe the sunlight is so bright today that we need little brightness. So with the help of diaphragm, we can control the amount of light. Condenser, it helps in focusing the light. Okay, the light is coming from all direction, but we need the light to hit the specimen, right? So condenser helps in focusing the light onto the specimen. Where is the specimen? It is on the stage. It holds the slides, or the specimen, you can say it holds the slides and the specimen. Now there are some kind of knobs. Okay, now these knobs helps us. It could be coarse focusing knobs or fine focusing knobs. So these kind of knobs helps us in adjusting stage. Okay, there's a distance between stage that holds lights and the objective lens. So at what distance should the objective lens work with respect to the stage? We use these kind of knobs. Okay, and fine focusing knobs, when you adjust your microscope using these kind of knobs, the movements will not be visible outside, but they are all internal. So these are very finely focused knobs that helps us in adjusting the stage and the objective lens distance. The other one is nose piece that holds all objective lenses. Now there can be different types of objective lenses as per their magnification. We're gonna study them in a later slide. Ocular lenses are the last part of the lenses for microscope. These are eyepiece that helps in viewing the image. Basically through ocular lenses we only see the image. Okay, now here, see the parts of microscope. Observe everything. We started with the light source, right? So there is your light source. Here is your light, and this is your basically diaphragm. This thing is your diaphragm, and that's how light, either if it, see, there's a bulb here. Okay, and if there's no bulb over here, there could be a light, sunlight, but there's a bulb, then there's a diaphragm, and then there's some called as condenser that helps in focusing the light. Okay, then we have the stage which has the slide, object nose piece which has objective lenses. This is your nose piece. Okay, this thing is your nose piece which helps in, I'm sorry. So here you have objective nose piece, and here you have nose. I'm so sorry. Here you have nose piece, and here you have objective lenses, and then it goes with ocular lens. See, here you can see there are two ocular lenses. So such microscope that have two ocular lenses are called binocular lenses because they have two ocular lenses. I hope it's clear. The same thing, you have the base. and you have such kind of arms, you call it arm, and here you can see the knob. So it helps us to maintain the distance between this objective lens and this stage that holds the slide. Okay, so this is your again ocular lens, objective lenses, your nose piece, your stage, your condenser, then your diaphragm, and then your here it's your light bulb. Now, this slide actually shows how the knobs are. So these are kind of knobs, and when you adjust them, this distance, the objective lens and the stage distance gets adjusted. See, now we are gonna talk about working of microscope, but before that, let's get a brief idea about it. When light rays enters into the stage or onto the specimen through condenser and the diaphragm, they all get focused onto the slide, and then they pass it through objective lens. They were first illuminated by the light source, then they pass through diaphragm, then they pass through condenser. Diaphragm helps in controlling the amount of light, then condenser helps in focusing. Here you can see they all are focusing the amount of light, and then these all focused lights when passes through slide, they get refracted, and many other phenomena takes place like the reflection, transmission, and then they pass through your objective lens. Okay, and then finally they hit ocular lenses through which we can see the image. See, now working, see, source illuminated, diaphragm controls the amount of light that passes through the specimen, then condenser helps us to converges the beam of light, or you can see the you can say focusing the amount of light. Then we adjust knobs because we need a proper distance between objective lens and the stage. Then light passes through the stage, it is coming from diaphragm, I'm repeating it again, it is coming from diaphragm, it is coming then to condenser, then it is hitting stage. Okay, it is hitting the stage that holds the specimen, and then it enters into objective lens magnify the object. Okay, that is the main function of objective lenses, that they magnify, and then image passes through the body tube that I showed you as an arm, and then we finally have ocular lenses which further magnify, because objective lens could be of magnification of 545x, you can say, and then ocular lenses are magnification of 10x. So you can say that the magnification we finally got is 450x. Okay, 45 multiplied by 10, 450 times, because it's x, we got the magnification. So one more time, passes through diaphragm, condenser, stage, then the objective lens which helps in magnify the object, then passes through the body tube, and finally hit the ocular lenses which further magnify and through which we see the final image. So that's it for this video, guys. Thank you so much. The other parts of the bright field microscope will be covered in the next lecture, so for that, stay tuned and subscribe for more. Bye bye, take care.